Greetings everyone, welcome back to this channel called Learn Bio with Janet. Today we will study an important compound called DNA, which is a type of nucleic acid. The most fascinating biological molecule must be DNA, or deoxyribonucleic acid. It has been called the molecule of life and the blueprint of life. DNA is a macromolecule that contains genes which determine the specific characteristics of an organism. A human being has about 25,000 genes, which contain instructions for the development and growth of a human being, as well as the maintenance of the living processes in the body. Genes are actually segments of DNA. Now, this DNA can be replicated or duplicated and passed down from parent to offspring through the gametes. So this is the first valuable thing that we receive from our parents. A normal human body cell, such as the skin cell, contains about 2 meters long of DNA that is coiled up to form the chromosomes. That's a lot of DNA. Of course, there are exceptions like the red blood cell that does not have a nucleus or chromosomes. Let's find out more about this molecule, DNA, that forms the blueprint of life. The learning outcomes for this lesson are as follows. Firstly, we must be able to list the elements in nucleic acids. B. Explain the structure of nucleotides, which are the basic units of nucleic acids. And each nucleotide contains a nitrogenous base, a ribose or deoxyribose sugar, and a phosphate group. C. We must be able to describe the structure of the nucleic acids, that is, the structure of the deoxyribonucleic acid and ribonucleic acid. Nucleic acids are one out of 4 organic compounds that are important and vital for the functioning of the human body. The other 3 organic compounds that are important are carbohydrates, lipids, and proteins. So what are nucleic acids? They are complex organic compounds which carry genetic information in the form of genes about protein synthesis in a cell. And they enable protein synthesis to occur in the cell. So in this way, the genetic information determines the characteristics of an organism, because the types of protein synthesized will determine the characteristics of the organism. There are 2 types of nucleic acids: deoxyribonucleic acid, DNA, which stores the genetic information in the form of genes in the chromosomes in cells. So it stores this genetic information for life in a person, and it can be passed down. This genetic information in the DNA can be passed down from parent to offspring, or can be inherited by the offspring or the child, because the DNA can replicate or duplicate itself, and it can pass down this genetic information through the sperm and ovum. Ribonucleic acid, or RNA, does not store genetic information for long. It actually copies the information, genetic information, from DNA when the cell needs to produce a certain type of protein. After copying the information, the RNA will then produce the type of proteins that are needed by the cell. So RNA uses the genetic information from DNA to synthesize proteins. It cannot be passed down from parent to offspring, not like DNA. Number 3, what's the difference, the basic difference between DNA and RNA? Firstly, is the type of sugar that is found in DNA and RNA. DNA contains a sugar known as deoxyribose sugar, hence its name deoxyribonucleic acid. And RNA contains another type of sugar called ribose sugar, hence the name ribonucleic acid. Let's discuss the structure of nucleic acids. A nucleic acid, DNA or RNA, is a polymer chain that is made up of basic units or monomers called nucleotides. As we know, a polymer is made up of basic units called monomers, repeating units of monomers that are joined together. So the monomer for nucleic acids are the nucleotides. Okay, this word is a bit difficult for students to remember, so you got to memorize it well. Now, so nucleotide is the basic unit of a nucleic acid. What is it made of? So it's made of three components: the phosphate group, PO4, 3 negative, pentose sugar, which is in the form of a ring. So it has 5 sides. It's a 5-carbon sugar and there's a ring that is 5-sided. Either ribose or deoxyribose sugar is the pentose sugar. Okay, for RNA, the sugar is ribose sugar, and for DNA, it is deoxyribose sugar. Both are 5-carbon sugars, and that's why they're called pentose sugar. Pentose means 5 carbon. Then lastly, there is one nitrogenous base. Only one base, but there are 4 existing bases or 5 existing bases. Okay, so the nucleotide can have either adenine as the base, or cytosine, or guanine, or thymine, or uracil. Okay, now uracil is only found in RNA, as we'll discuss later. But we need to know all the names of these bases. They're called nitrogenous bases because they have nitrogen, the element nitrogen, in their molecular structure. Number 6, the formation of nucleotides. So the phosphate group, sugar, and base of a nucleotide are all combined together through the process of condensation. Remember condensation? It is a process in which the smaller molecules like phosphate group, sugar, and base are combined together with the elimination of water molecules or the removal of water molecules to form a more complex molecule, such as the nucleotide. Now, chemical composition. Nucleic acids are formed from the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. So, the short form is CHOM. Remember that. We will first discuss the DNA molecule, followed by the RNA molecule in the next lesson or video. So the structure of DNA is that it is a double-stranded nucleic acid, which is a long and complex macromolecule or polymer. It is made up of 2 strands of polynucleotides twisted to form a double helix. Right, the word double-stranded means that it has 2 strands of polynucleotides. So each strand consists of many nucleotides. That's why we call it polynucleotides. Many nucleotides are joined together in each strand. Okay, millions of nucleotides. But there are 2 strands, separate strands. And these 2 strands are held together in the center, right? Then they are twisted to form a double helix. So they are like coiled around each other, twisted or coiled around each other to form a double because there are 2 polynucleotide strands. Helix means a spiral shape like this. Okay, it looks like a ladder, but a spiral ladder. Location. The DNA is found in the chromosomes in the nucleus of a cell. So a large part of the chromosomes, okay, are the DNA molecules, which are very long, and they are coiled up tightly in the chromosomes. So as I said, the human cell contains about 2 meters long of DNA, each cell, and that's about 6 feet or more. Anyway, if you look at the molecule of DNA, it's very unique with its double helix shape, and it's also very beautiful. Right, let's go on to the function. The DNA forms the genetic material in an organism. All right, So a long DNA molecule actually has many segments or sections that forms the genes. A segment or section of DNA forms a gene. A gene determines a characteristic of an organism such as hair color and blood type. So we have genes for hair color, we have genes for blood type, genes for height, to determine even the height of a person. So a gene is the basic unit of inheritance, meaning it can be passed down from parent to child in the sperm and ovum. Okay, so father will produce the sperms, mother the ova, and if a sperm were to fertilize an ovum, the genes in them will be passed down to the zygote that's formed and to the new organism that's formed. Okay, now DNA contains many genes with information to make different types of proteins. Okay, so each gene has the instructions to make a specific type of protein. And what are the proteins that can be made from the instructions given by the genes? So all types of proteins, for example, hemoglobin, enzymes, antibodies, hormones. Think of the word H, E, A, H here, right? For the different types of proteins that our body can make. Now, these proteins that are formed then determine the characteristics of a person. So genes help to determine the characteristics of a person through the production of the different proteins. To explain the structure of DNA, we first have to understand the structure of the basic unit of DNA, and that is the nucleotide. So many molecules of these nucleotides are joined together to form the large DNA molecule, which is a polymer. So let's look at the structure of one nucleotide, as shown here in this picture. It looks a bit like a hand with the thumb, the palm of the hand, and the fingers. Okay, now this part, this component that juts out here is the phosphate group, which consists of the phosphorus and oxygen atoms. And then we have the pentose sugar in the middle, which is a 5-carbon sugar. For DNA, it's called the deoxyribose sugar. For RNA, it's ribose sugar. So here for DNA is deoxyribose sugar as the pentose sugar. And this sugar has a shape like a pentagon also, which is 5-sided shape. Then at the other end, we have the nitrogenous base, only one nitrogenous base that is attached to the pentose sugar. So this nitrogenous base here can be any one of these four, but only one. It can be the nitrogenous base called adenine with the symbol A, guanine with the symbol G, cytosine with the symbol C, and thymine with the symbol T. So all these are called nitrogenous bases, but only one is attached to each pentose sugar in a nucleotide. That means that if you have a group of nucleotides, the phosphate group and the pentose sugar group will be alike in all of them, and it will be the nitrogenous base that differentiates them. So there are 4 types of nucleotides. The first type will have adenine as the base. The second nucleotide will have guanine as the base. The third nucleotide will have cytosine as the base, and the fourth one will have thymine as the base. 4 different nucleotides. Here is an acronym to help you remember the 3 components of the nucleotide. The 3 components are phosphate group, sugar, pentose sugar, and the nitrogenous base. So we just write down base. So remember, please stay back. P for phosphate group, S for sugar, B for base. Or another acronym, you can choose which one you want, is phone Sue the baker. Phone, PHO, phosphate group, Sue for sugar, baker, BA for base. Especially if you crave some cakes, sometimes you can phone your baker and ask her to make you a nice carrot walnut cake, for example. Okay, so phone Sue the baker to order a cake from her. Now I hope that you will be able to remember the components of the nucleotides through these acronyms, because many students have a problem even remembering this name, nucleotide. Okay, so try to remember the word nucleotide is the basic unit of DNA and also RNA. And then the three components of the nucleotide has to be memorized. Okay, it's important for the exam questions, especially the objective and structured questions. Here is some further information concerning the structure of the nitrogenous bases of DNA, which is adenine, thymine, guanine, and cytosine. These structures were shown in the textbook, so we have to know basically the shape of these molecules. For example, adenine comprises two shapes that are combined together. One is a hexagon shape and the other is a pentagon shape. Okay, joined together. Same with guanine. It has a hexagon shaped ring and a pentagon shaped ring. Okay, so but there are some minor differences between these two, the adenine and guanine. Notice that all the bases have nitrogen in them. That's why they are called nitrogenous bases. Apart from nitrogen, they have carbon and hydrogen, and some of the bases have oxygen atoms also. Now for thymine and cytosine, they both have quite similar structures, and the shape of the molecule is a six-sided shape like a hexagon. Okay, all these shapes were shown in the textbook, so we have to basically recognize that the hexagon shape is for thymine and cytosine, and this shape, which is a pentagon and hexagon, is for adenine and guanine. A simpler form will be to represent adenine by any shape shown here and thymine by a matching shape. Okay, because adenine always pairs or bonds with thymine, as we'll see later. So the two shapes must be complementary or matching each other. You can choose any shape, but it must be matching. The shape of A to T must be matching. And G and C are complementary bases. They will pair up together or bond together. So the shape for G must be complementary to the shape for C. So these are the simplified forms to represent the nitrogenous bases. Here are some further details about the molecular structure of a nucleotide. This is the molecular structure of the nucleotide. So it has three parts: the phosphate group with the phosphorus and oxygen atoms seen here. This is the pentose sugar, okay, with the pentagon shape ring, and it has 5 carbon atoms, 1, 2, 3, 4, 5, with an oxygen atom in the center here. So this pentose sugar is deoxyribose and is linked to or joins to the nitrogenous base, adenine. Okay. However, this diagram need not be memorized. In the textbook, we use this diagram, simplified diagram, to represent the nucleotide. Phosphate group represented by a circle, then you have the sugar represented by a pentagon, and then you have the base that's joined to the sugar. So the sugar is in the middle. Remember that. So remember PSB for phosphate group, sugar, and base. That these are the three components of the nucleotide. Okay, of a nucleotide. And the acronym, please stay back, or polis suka batik. Now let us discuss how the nucleotides are joined together to form the complex molecule of DNA. This diagram shows you the first nucleotide. Now the pentose sugar of this nucleotide will form a bond with the phosphate group of the next nucleotide. Pentose sugar with phosphate group of the next nucleotide. Okay, so a bond is formed between them, And then similarly, the sugar for the second nucleotide forms a bond with the phosphate group of the following nucleotide. In this way, a lot of nucleotides are joined together where the sugar of one nucleotide forms a bond with the phosphate group of the next nucleotide. So when the nucleotides join together, for example, millions of nucleotides can join together to form a long chain or strand, we call that chain or strand a polynucleotide strand. Poly means many nucleotides joined together, okay, to form a long strand. So this is one polynucleotide strand. Here is our polynucleotide strand, one of it. But DNA consists of two polynucleotide strands. So how is the second strand joined to this first one? First, we just label the components of the nucleotide, the phosphate group, pentose sugar, and nitrogenous base. Now, the second polynucleotide strand is joined like this to the first one. Hydrogen bonds are formed between the bases from the two strands. Black lines represent the hydrogen bonds. Adenine forms two hydrogen bonds with thymine, and guanine forms three hydrogen bonds with cytosine. And the sequence of bases does not necessarily have to be A, G, C all the time. It can vary or change according to the gene and the genetic code on the genes. Okay? Now, so adenine and thymine are complementary bases because adenine can only bond or pair with thymine, and guanine always bonds or pairs with cytosine. So we can show this that they are complementary bases by showing it through the shapes. The shapes must match each other. So adenine and thymine are complementary bases. Guanine and cytosine are complementary bases. Now, adenine cannot bond with cytosine. You can see the shape does not match because they are not complementary bases. Okay, as an example, guanine cannot bond with thymine. They are not complementary bases. Now, let's look at the polynucleotide strand. Okay, the molecules here on this side, the pentose sugar and phosphate group. Notice that the pentose sugar is pointing downwards on this side. The pointed end is pointing downwards, but for this side is pointing upwards. So we say that the two strands are parallel, but since one is pointing downwards, the other one pointing upwards, we call them anti-parallel. Okay, the two strands are anti-parallel. That means they run in opposite directions to each other. Okay, and then we must know how to draw this diagram. So in the exam, one part has been shown before and the student had to draw the other side of it, fill it up. So the bases must match first of all, and then you must draw the pentose sugar facing the other way compared to the first polynucleotide strand. Okay, so if you're asked to draw the structure of DNA, you can draw it like this. Thus, it's good if you can draw this diagram into your exercise book as practice after learning this lesson. So here you have the molecule, the two strands of DNA joined together at the bases by the hydrogen bonds. Now sometimes in a picture like that, you may be asked to circle the one nucleotide, circle just one nucleotide, especially for structured question. So this is one nucleotide made up of a phosphate group, a pentose sugar, and a base, nitrogenous base. Okay, now we look at the overall structure of the DNA molecule. we can see that it looks like a ladder. The two sides of the ladder are made up of the sugar and phosphate groups. Sugar, phosphate groups. Okay, the sugar and phosphate group will alternate with each other. So we call this the sugar-phosphate backbone of DNA. In the center are the so-called rungs of the ladder, which are made up of the base pairs, where adenine will always bind with thymine and guanine with cytosine. Okay, bound together by hydrogen bonds. However, this is not the final shape of the molecule because DNA has a double helix structure. Helix means like a spiral shape. So imagine that this molecule, just in your imagination, give the molecule a twist. Let's twist. And finally, you have the double helix structure of DNA. Okay? So a helix is a spiral shape or a coiled shape, like this. And here you can see the sugar phosphate backbone is the orange part here. It's for the first strand, and the second strand also has the sugar phosphate backbone. And in the center are the rungs of the ladder, which are actually the base pairs, where adenine will always bind with thymine and guanine with cytosine only. So this is the double helix structure of DNA, and we have to remember this term, double helix structure of DNA, which was discovered by a pair of scientists long ago. Their names are Watson and Crick, and they won the Nobel Prize for being able to explain the structure of DNA, for discovering the structure. Now sometimes the DNA molecule can be presented by this structure shown here. It is a simplified form of the DNA molecule, where the pink part represents the phosphate group, the yellow part represents the pentose sugar, and the bases are again in the center. So these are two polynucleotide chains, okay, with the sugar phosphate backbone, and the bases form the hydrogen bonds between each other in the center. Okay. Then sometimes you will be asked to just circle one basic unit called the nucleotide. Okay, so you can circle like this and label this as one nucleotide. This is another version showing the structure of the DNA molecule, and it's something similar to what you have in the textbook, so I will have to explain this to help you understand. Okay, so in this diagram we still see the nucleotide here joined together to form a long polynucleotide strand. Okay, so this is the sugar, this is the phosphate group, which is joined to the sugar, and then phosphate group, sugar, and so forth. On this side is the same. This is the other polynucleotide chain or strand, but it's facing downwards. The pointed end of the sugar is facing downwards, whereas this is upwards. Okay, so we say that these two strands are anti-parallel to each other. And here again are the bases, right? A, G, and T. Right, so the difference between this version and other versions is the details, okay, found here, the details found here, where the bases have the shapes that look like the real shapes that they have. Okay, so adenine comprises a hexagon shaped ring and a pentagon shaped ring that is combined together. Alright, like the actual case as you have seen for the molecule of adenine. And guanine too, right, has a hexagon and a pentagon shaped ring to represent its molecules here. However, this is not the actual molecular formula for the bases because they don't show the nitrogen atoms and some other atoms. Okay, so for thymine and cytosine, we can see the hexagonal or the hexagon shape here. You can see the hexagon shape here. The T and the C are quite similar in shape. Okay, however, G will only bind to C and vice versa. A will only bond with T or vice versa. Okay, so G will form 3 bonds with C. Guanine will form 3 bonds, 3 hydrogen bonds with cytosine, whereas adenine will form 2 hydrogen bonds with thymine. Okay, so the red lines represent the hydrogen bonds. And that's all that you need to understand here. So if you see that there are 2 bonds between the bases, it must be adenine and thymine, this pair of bases. If there are 3 bonds, it must be the bond between guanine and cytosine. So this pink part here is the sugar phosphate backbone, and also the blue part here for the 2 polynucleotide chains. And if you're asked to draw or circle 1 nucleotide molecule, then this is it. Okay, 1 phosphate group, 1 pentose sugar, and 1 base. Mix up the nucleotide. Now, in the DNA molecule, which is the most important part? Is it the phosphate group, the pentose sugar, or the base? Yes, the most important part are the sequence of bases in the center part of the DNA molecule. The sequence of bases, for example A, G, and C, forms the genetic code for the synthesis of proteins. So AGC may represent an amino acid that must be used in the formation of a specific protein. It's interesting to study how proteins are formed by using these genetic codes, and we'll do that in the following video on the production of proteins. Here is an exam question that we can discuss, and the model answers are given below for you to copy. Question 1. Explain the structure of DNA. 10 marks. So this is the model answer. Firstly, we must state that the basic unit of DNA are the nucleotides, as shown here. And each nucleotide is made up of 3 components: a phosphate group, a pentose sugar called deoxyribose sugar, because it's for DNA, and a nitrogenous base, which can be one of the following: adenine, thymine, guanine, or cytosine. Next, state that the many nucleotides are joined together to form a long polynucleotide strand. To join together, the pentose sugar of each nucleotide bonds to the phosphate group of the next nucleotide. Okay, and in this way, the nucleotides are joined together to form the polynucleotide strand. Fourthly, 2 different strands of polynucleotides are bonded to each other by weak hydrogen bonds between their bases to form the DNA molecule. Okay, so this is one polynucleotide strand, this is the other one, and they are bonded to each other by the weak hydrogen bonds between their bases. Next, mention that the 2 strands are antiparallel. They run in opposite directions, one facing upwards and the other one downwards. To explain the structure of DNA, we have to mention the double helix. So how is it formed? So the DNA molecule consists of 2 polynucleotide strands that are intertwined in opposite directions to form a double helix. So this is strand number 1 and this one is strand number 2. So they are intertwined, meaning they are coiled around each other, okay, in opposite directions. One goes this way, this one goes the other way, to form the double helix, which is a spiral shape. Finally, to answer the question, explain the structure of DNA, we can state about the complementary bases. Okay, the nitrogenous bases on one polynucleotide strand here are matched to their complementary bases on the other polynucleotide strand. The base adenine always bonds or pairs with thymine, and the base guanine always bonds or pairs with the base cytosine. The bases are bound together by weak hydrogen bonds. And that explains the whole structure of DNA. Each point is 1 mark, okay, like number 8, 1 mark, number 9, 1 mark, and so forth. Lastly, let's discuss the location of DNA in a cell. It's very simple. We'll zoom in from the bigger structures to the smaller structures. So in a cell, there is a nucleus. And in the nucleus, there are many chromosomes. For example, a normal human body cell, such as the skin cells, will contain 46 chromosomes. Each cell will have 46 chromosomes. Right, so now, if we were to enlarge one chromosome, in this case it's shaped like an X, you will see that it contains a very long strand of DNA that is coiled up inside the chromosome. So this long strand of DNA contains the genes. Now, what is a gene? A gene is just a segment of DNA that has the information for the synthesis of a particular protein. So the genes are arranged linearly along the length of DNA. For example, here you may have one gene and then another gene further down and so forth along the strand of DNA. So in the genes, where is the important part that stores the genetic code with the information for the synthesis of a specific protein? It is the sequence of bases in the center of the DNA molecule. The bases being adenine, thymine, cytosine, and guanine. So they are arranged in an order that's very specific, and it must be arranged in a specific order to code for the various amino acids that need to be joined together in the right sequence to build a protein molecule. We'll study more of this in the next video, that is the synthesis of proteins. Here are the notes for the explanation given in the previous slide. The location of DNA and genes in a cell. So in a cell, there is a nucleus containing many chromosomes. Each chromosome is made up of a long strand of DNA, which is made up of sections of DNA called genes. Each gene is made up of many nucleotides, and each nucleotide consists of a phosphate group, pentose sugar, and base. Thus, a gene contains a sequence of nitrogenous bases such as adenine, cytosine, guanine, ACG, then CCA, and so forth. Each sequence of bases is a code or an instruction to make a specific protein in the cell, right? So that ends the video for this part, and the structure and function of RNA will be explained in the next video, as this video is already quite long.